DNA computing and molecular programming is a subset of nanobiotechnology that uses the biochemical properties of DNA to perform certain operations. DNA computing at this time is a highly academic pursuit whose real-world applications have yet to be fully exploited. The aim of this project was the production of a DNA-based molecular automation which binds to glucose with the possibility of cleaving insulin to reduce blood sugar using DNA. Achieving this goal would greatly increase the knowledge in this relatively young field and demonstrate a practical use of this previously abstract technology. Computational chemistry methods have been used to decrease the research costs while allowing a novel approach to oligonucleotide aptamer generation. In order to allow the study of many oligonucleotides to find one with the appropriate properties, the workflow emulated the SELEX procedure. This methodology could potentially be used for the production of any DNA-based aptamer. Initially, a large amount of oligonucleotides was generated computationally in order to create an oligonucleotide library. This library was then screened against glucose to test for an interaction between the DNA aptamer and glucose. The strength of the interaction between glucose and the DNA aptamer was investigated in order to select aptamers depending on their ability to bind reversibly with a sufficient interaction. The selected aptamers were then produced experimentally in order to test their interactions using surface plasmon resonance and verify the computational data. Finally, a fluorophore quencher was added to the DNA strand in order to functionalize the aptamer as a glucose biosensor. The main techniques used in this investigation was a multitude of computational chemistry programs. Computational chemistry aims to model the behavior of atomic and molecular systems. This allowed me to investigate the interactions between a large amount of oligonucleotides at a low cost. In order to speed up result turnover time, a computer cluster of 20 computers was used. Accelerary's Creation Studio was used to generate the oligonucleotide library. In order to reduce computational error, a select amount of oligonucleotides from Accelerary's was fed into Gaussian, with a geometry optimization run on them. This was to confirm that the oligonucleotides generated were of sufficient quality to enable further investigation. Further investigation was done using Autodock to simulate ligand-receptor docking. The oligonucleotide library was then used in Autodock to investigate interactions between the glucose and the DNA aptamer. The data was then investigated using PyMol in order to visually analyze the conformational change around glucose. A select number of oligonucleotides were used in Gaussian for a single-point energy calculation. From this data, 2 aptamers were selected that had the most promising results. The surface plasmon resonance machine was prepared using the CM5 chip, where 1-ethyl-3,3-dimethyl-aminopropyl carbonamide and N-hydroxysuccinimide was flowed over the chip, which bound to the dextrin matrix. The ligand was then flowed across the chip, which in this case was glucose. The glucose bound to the dextrin chip covalently. If there was any glucose that did not bind covalently to the dextran strip, it was removed using an ethanol amine wash. Afterwards, the analyte is flowed over the chip. This produces a response unit. A glucose biosensor was created by the addition of a 6-fluorescent fluorophore and an Iowa black FQ quencher on either end of the DNA strand. A photospectrometer was used to detect the fluorescence output of the oligonucleotide biosensor. When the DNA aptamer is on its own, the fluorophore and quencher distance is at its greatest apart. However, when glucose is added to the mixture, the conformational change caused by the glucose aptamer to the glucose causes the fluorophore and quencher to become closer to each other, thus reducing the fluorescence output. A larger concentration of glucose should produce a larger decrease in the amount of fluorescence observed to the point of complete saturation. Initially, a large amount of oligonucleotides were produced with a range of base pairs from 10 to 25. It was found that the higher the amount of base pairs, the higher amount of computational time was required, and as such, a lower amount of base pairs was generally favored. Upon inspection of the Autodoc output files, many of the oligonucleotides presented had multiple configurations, as demonstrated here. The most stable energy-wise conformation was chosen from the multiple configurations. A large range of configurations were found, as demonstrated by these nucleotides, based upon the single-point energy calculations and the Autodoc outputs in PyMol. I chose these two oligonucleotides. The oligonucleotides were prepared for the surface plasmon resonance machine by serially diluting the oligonucleotides into different molar concentrations. The molar concentrations were then flowed across the CM5 chip prepared, as mentioned earlier. Initial investigation into the dilution results indicate that there is a response between glucose and the DNA aptamer. However, it is quite small. The difference between the 12 base pair long oligonucleotide and the 10 base pair long oligonucleotide is approximately 700 response units. When the response was compared against a control chip, it was found that there was indeed a change in response, demonstrating that there could be an interaction. However, this data was inconclusive, as the amount of response was quite minimal. As such, the fluorophorin quencher investigation was used to confirm an interaction. The low response could be due to the small size of the oligonucleotide, only being about 10 base pairs long, or due to the CM5 chip taking a less than desirable amount of glucose onto the dextran strands. A further possibility could be due to the fact that the interaction between glucose and the DNA aptamer is of a hydrogen bond nature, as the computational data lends this suggestion, as well as the SPR results. Glucose was added to the DNA aptamer with the fluorophorin quencher added to either end at differing concentrations, 10 micromole, 20 micromole, and an excess of glucose. The response received from the photospectrometer indicated that there is a change in the fluorescence output of the oligonucleotide when exposed to higher concentrations of glucose. However, this response is very small. This is likely due to the aptamer itself being very small in terms of distance. This could be refined by increasing the number of base pairs in the aptamer, or adding extenders to either end. In conclusion, a successful DNA-based glucose biosensor was produced with a low sensitivity. However, the sensitivity could be greatly increased by further redesigning of the oligonucleotide, either by increasing the number of base pairs in the computational stage, or by simply adding extenders to the oligonucleotide in the production phase. This oligonucleotide production methodology could be easily applied to multiple different molecules in order to create a low-cost aptamer production. However, the requirement of a computer cluster is highly recommended, as computational times can often exceed multiple days on a single computer when calculating multiple jobs. Aptamers could be further functionalized by the addition of DNA spiders onto the outside strand in order to recognize glucose in the bloodstream and cleaving insulin in order to reduce the chance of hyperglycemia.